Hi, my name is Jeff Becker, and this talk is about Tropilaelaps mites, the next invader. And this is for my GPMB Master Candidate project. I'm going to talk about why Tropilaelaps is a greater threat than Varroa, how it spreads, and how it can be detected in honeybee hives, how it can be managed, and unfortunately, there's very little good news, but there is some. So what do I mean by next invader? Like the European honeybee itself, the most troublesome pests for managed honeybee colonies came from overseas. Except for the tropy mite, which has not yet reached the US, these should be pretty familiar. The wax moth most likely arrived in the US with the first managed bee colonies and was for a long time the biggest challenge beekeepers faced. The Rula fly, while not a major concern, is of interest because some researchers think it might be mistaken for the tropy mite, even though it is quite a bit larger. Tracheal mites caused significant colony losses when they first arrived in the US in the mid-1980s. Over time, the threat has lessened, and it's not entirely clear why this is the case, whether it's natural resistance, the effect of other treatments on the tracheal mite, or both. Varroa destructor is, as we all know, the worst pest beekeepers have to deal with. I'll be comparing Tropilaelaps mites to Varroa mites in order to show why the Tropilaelaps mite is the bigger problem in areas where both exist. The small hive beetle arrived in the US in the late 1990s, originally from sub-Saharan Africa. It is a bigger problem in the warmer southern states, but it's a problem elsewhere. And this is what they look like. So Tropilaelaps mite, it's quite a bit smaller. It's an ectoparasite that jumped from an Asian honeybee species like Varroa from Apis cerana, Tropilaelaps from several related giant honeybee species. Because European honeybees did not co-evolve with these mites as the original hosts did, I mean both Varroa and Tropilaelaps, they have not evolved similar defenses as the original hosts. The resulting infestations have devastated European honeybee colonies in Asia and every place else these mites have spread to. So let's look at these original hosts. The original host of Varroa was Apis cerana, and the original host of Tropilaelaps was a variety of Asian giant honeybees, and there are different species of Tropilaelaps that have been hosted by different Asian giant honeybees. So of the 4 known species of Tropilaelaps mites, only 2 are able to parasitize European honeybees, and these are T. mercedisi and T. clarei. T. clarei is only known to exist in the Philippines, as it was originally hosted by the Philippine giant honeybee. T. mercedisi has a wider geographic distribution and is generally considered a greater threat. So for the remainder of this talk, when I talk about Tropilaelaps, I will be referring to T. mercedisi. I may also refer to them as tropy mites or just T mites. So Tropilaelaps was originally present in the range of the giant honeybees, which is shown in the polka dot fill. It has since spread to Papua New Guinea, China, South Korea, Uzbekistan, and most recently into Soviet Georgia and a province of Russia. Tropilaelaps has been gaining increased attention worldwide because of its recent appearance so close to Europe, in particular Ukraine, where there's quite a bit of beekeeping. Tropilaelaps has repeatedly defied expectations and has survived in conditions previously thought to be fatal for them, in particular the spread into temperate regions in Korea and China, where there was typically a natural brood break, proved that this is not necessarily a limiting factor for the spread of this mite. So this map shows the spread of Varroa mite over a period of many decades. Australia was the last continent to be invaded, so no one knows how entered Australia, but it was detected in June of 2022 in sentinel hives at the port of Newcastle, New South Wales. They mounted a huge effort, a 14-month-long attempt to eradicate varroa, costing millions of dollars and the destruction of more than 14,000 hives. It was decided that eradication was not possible, it wasn't working, and they would have to manage varroa like everyone else. Because of experiences like this and this one, it is understandable that many experts can consider the continuing spread of Tropilaelaps as inevitable. So why is Tropilaelaps such a big problem? So Tropilaelaps reproduces much faster, even though their life cycles look similar. There are differences that lead to Tropilaelaps reproducing much faster. So here are some of the details. So the founder's mite starts laying eggs 10 hours after capping of the brood cell. Varroa, the founder's mite, takes about 60 hours after capping before she starts to lay. The Tropilaelaps mite lays eggs at about 24-hour intervals. The Varroa mite lays eggs at about 30-hour intervals. In addition, the Tropi mite's reproductive success is generally higher. So the Tropilaelaps dispersal phase lasts only 1 or 2 days, averaging about 1.3 days. This has major implications as far as how these mites can be managed. The Tropi founder is sexually mature after this short period of time and can start reproducing right away. The Varroa dispersal phase is longer. It's 4 to 6 days, and the founder's mite needs about 5 days to reach sexual maturity. So there's an additional delay. Because of very rapid population growth, Tropilaelaps can kill a colony in a matter of months rather than a year or more for Varroa. So studies have shown that Tropilaelaps infestations can grow very rapidly. So practically nothing to 15% in just 3 brood cycles, or about 63 days. A higher proportion of Tropi mites produce at least one progeny, meaning they get more children. And amazingly, when there are more founder's mites in a cell, in a single brood cell, they each produce more offspring. So what this means is that the worse the infestation gets, the faster the population can grow. And to make matters worse, not only do they build their populations much faster, but they do more damage. So Tropilaelaps mites feed on the human lips of larvae and pupae, causing reduced weight and reduction in their overall lifespan. Varroa feeds on adult bees as well, so they spread out their damage somewhat. Tropilaelaps opens to multiple feeding sites, and this is pretty significant because they form wounds that persist through the transformation from pupa to adult. Varroa, on the other hand, they tend to open just one communal feeding site. And both are virus vectors. Tropilaelaps happens to be a really good vector of deformed wing virus, which is particularly damaging. As far as detecting Tropilaelaps, so far, mostly it happens after a colony has actually died, and then they'll send in a sample to a lab, a sample of brood to a lab. It'll be frozen, and then individual cells are opened, and the brood is inspected for mites under magnification. Recently, DNA testing has become available at the National Agricultural Genotype Center, and so that would be another fairly reliable way of determining if a colony is infested with Tropilaelaps. A study was published just last year by Gill and Associates that evaluated monitoring methods being used by the National Bee Unit in the UK. They found the following: that alcohol wash does not work. The CO2 wash does not work. These tend to not dislodge Tropi mites from adult bees. The method involves hitting brood frames against a solid surface to dislodge the mites. It was designed specifically for determining if T mites are present in a hive. Not only is it not very good at detecting the presence of T mites, it tends to injure or kill honeybee larvae. Brood removal using an uncapping fork is poor at detecting Tropilaelaps because it damages the pupae and the mites get lost in the goo. As part of this study, they developed a refinement to this method using fine-nosed tweezers. The pupae remained intact better, and this performed better as a test for finding Tropilaelaps. Screening high floor debris by floating the debris in either alcohol or ethanol or water doesn't work very well. It was thought that T mites floated in ethanol, but they don't, and they do float in water, but so do other things. So it just makes things hard to find. So another refinement was tested using a sticky board and a shorter period of sampling the debris drop onto the sticky board, and that worked a bit better with good lighting and magnification. So this is what the results of the study looked like, and actually the surprise winner here is a sugar shake was pretty effective at determining if a colony is infested with Tropilaelaps mites. Uncapping with and removal of brood with tweezers turned out to be the best in this study. Other methods performed rather poorly. But as a result of this study, and this happened fairly quickly, the National Bee Unit in the UK is now using the two tests, the sugar shake and uncapping with tweezers, in their the hives they're using to monitor for Tropilaelaps entering the country. While trying to help Georgia beekeepers with their Tropilaelaps invasion just last year, 2024, Ustinov and Associates also decided that a more efficient method of determining if a hive was infested was needed. So the method they came up with uses wax hair removal strips and a video camera. So this and the following images are from a video on Tropilaelaps info that demonstrates this method. If you want to see the full video, please go there. And the way this works is first the wax strip is applied to cap brood. Then while recording, the strip is removed rapidly, at which point the mites rush out of the cells and start running around. And by watching the video on a large monitor after the test is actually done, it's possible to count those mites. This method identified infested colonies in over 90% of the cases and failed only for very low infestation levels of less than 0.5%. They also found a strong correlation between mites counted in the video and mites remaining in that section of brood as counted in the laboratory. So this method could also be useful in determining the level of infestation. This is a video that shows what might be getting captured when the mites start running about. And so these little white specks that are running around, they're tiny, they move fast. Those are Tropilaelaps mites. So how is it? So another study was done to determine how Tropilaelaps was surviving broodless periods in temperate climates. And there are a number of ways that they can survive. They survive about 4 days on live adult bees, Apis mellifera honeybees, 6 days on decomposing pupae, and 7 days on decomposing adults. The tropy mites were observed spending the majority of their time on dead pupae and dead bees, and appeared to be feeding on the exudate created during decomposition. Tropilaelaps can also spread on adult honeybees. So this study was done using funnel traps to catch adult bees leaving the hive, and they proved that Tropilaelaps can move between colonies on adult bees. So nearby apiaries can become infested even without a beekeeper moving materials between those apiaries. this is pretty huge. A certain, but they also, some good news. A certain threshold of infestation was required before T-mites are likely to attach to adult honeybees. This study did not determine that threshold, but I'm assuming a subsequent study will. So keeping infestation levels low might help slow the rate of spread of these mites. So what are ways that Tropilaelaps can spread? So some of these were thought to be not a problem, like live honeybee sales, but since it's found that tropy can survive for a certain period of time on live bees and dead bees, both of which may be present in a package, or other forms of live honeybee cells like queens with attendants, they could possibly spread Tropilaelaps because quite often these bee sales happen over a short period of time between getting the package set up and it arriving at its destination. Likewise, sale of used beekeeping equipment that contains dead brooder bees, as long as it's moved relatively quickly, could be a problem. As always, migratory beekeeping is a great vector for pests and diseases, including tropy mites. And feral colonies or swarms on shipping containers or other vessels could spread tropy mites far and wide. And this picture on the lower right, a swarm has entered a container through a keyhole. So what are some ways that Tropilaelaps can't spread? So a study was done looking at some common hive products to see how long tropy mites could survive on them. There's only 3 days on some fairly common things like honey or pollen, royal jelly, and sucrose. So this implies that T-mites are not likely to spread through these common trade goods. So IPM for Tropilaelaps is a work in progress. There is no defined IPM for T-mites in the US. So prevention. So many countries have monitoring programs in place, including the US. The hope is that if the mites can be detected early enough, then they can be eradicated. Unfortunately, there doesn't seem to be any way to eradicate them other than perhaps burning hives, since chemical controls have been shown to not result in complete eradication. So it may be wishful thinking to think that we can allow them into the country and then eradicate them. But in the US, the USDA conducts a national survey so that honeybee beekeepers can sign up for and participate in, and they also have labs where samples can be sent to check for T-mites. The National Bee Unit in the UK monitors a number of hives. They have both some government and some volunteer sentinel hives that are monitored for tropy mites, and a similar program in Australia. So we do know that there are restrictions on honeybee trade goods, such as getting packaged bees in and out of Canada and Queens and whatnot. However, Canada does do trade with Ukraine, and Ukraine's right on the firing line for the next invasion of Tropilaelaps. So it remains to be seen whether additional restrictions will be put in place. Well, one hopes that they will be. Brood breaks have proven to be a very effective cultural control for Tropilaelaps. And in this case, a brood break alone, so this is on the lower right, for day 60, the first column is showing just a brood break. The middle column where it looks like there's nothing there, that's brood break plus formic acid, and the amount was so low that it actually did quite well. And then brood break with oxalic acid also did rather well. But this in this study, while brood breaks worked quite well, they did not achieve complete eradication, as I just mentioned. So now for hygienic behavior in bees, hygienic behavior shows that there is some promise that this will help in managing Tropilaelaps. So there was a study in 2001 where it wasn't actually part of the study, but this was an observation made during the study that workers appeared to be detecting and removing parasitized brood that was infested with Tropilaelaps. Again, in another study in 2017 observed more or less the same thing. And a study conducted in Thailand where the bees have some hygienic behavior showed that the hygienic response significantly reduced Tropilaelaps reproductive success. So while none of this is conclusive, it gives us some hope that improved hygienic behavior through queen breeding will produce results for both Varroa and Tropilaelaps. As far as physical controls, heat treatment proved to be somewhat effective. 106 degrees for 160 minutes led to an 85% decline, which is not good enough by itself, but in combination with other controls, such as chemical controls, it could reduce the amount of chemical needed. There are no known biological controls for Tropilaelaps. So some bad news is that most of the synthetic miticides we use for Varroa don't work against Tropilaelaps. The only one that showed some effectiveness was Apivar at about 64%. And the researchers, their conclusion of this number was that it shows that T mites are already developing some resistance to Amitraz. 64% is not good enough to be used as a control. Researchers think that the short dispersal phase for tripping mice is partly responsible for why these chemicals don't work. So formic acid, consistently effective against Tropilaelaps. Formic Pro, in the study, Formic Pro was tested and it worked quite well. And liquid formic, which was used in Asia, they soaked wood blocks with it. And while it's a bit ad hoc and not carefully metered, it also proved reasonably effective. Formic acid also performed other treatments. It's actually worked the best, better than the next best, which was powdered sulfur, which is used in Asia. We don't tend to see it here. And then things like the hops acids work no better than actually nothing. So the main problem with formic is that it does have temperature limit. So if it happens during a time of year when it's too hot to use, then that could be a problem. So my hope is that new metering technology will be developed. And there was an article recently that showed that there is a device being produced for metering different miticides. So hopefully that there will be technology produced that will allow formic to be used at different temperatures than what we're currently restricted to. And as always, rotating treatments is a good idea. So what do we say about this? We cannot predict how long it'll take Tropilaelaps to get here. If we're lucky, it won't be in our lifetime, and it would be a sad day if we look back on today and think, well, those were the good old days before Tropilaelaps. The rapid growth of Tropilaelaps populations means monitoring will need to be more frequent, so faster, more efficient monitoring methods are needed. In Asia, treatments for Tropilaelaps generally work against Varroa, so they focus on Tropilaelaps, and Varroa gets handled in the process. So this is likely to be our life here if Tropilaelaps invades the U.S. So sorry for the gloom and doom. That's the way it looks, and here are my references. And thank you for watching.